Welcome back. Let's continue to look at insulin as a product, as we looked in the last video. So we're going to look at, as is mentioned, a little bit more about the engineering of insulin production. Okay, so as a reminder, insulin is a hormone, a peptide, a small protein, which actually helps transport glucose from the bloodstream into the cells. Okay. So, as I mentioned in the last video, insulin was first extracted from tissue, animal tissue, animal pancreas, for example. And that was fraught with problems because you were taking a foreign protein injected into humans, and that causes immune problems, right? So then the Sanger discovered or determined the sequence of insulin, the two chains of two polypeptides linked up with this disulfide bond, as they call it. He actually did the experiment to discover what that sequence was. By that I mean, what is the insulin made of? Insulin is a polymer. They're made of individual units that are linked up together. He determined what that sequence was. That was the experiment in 1955, for which he got a Nobel Prize several years later. So, what insulin is, it's a polymer made of individual amino acids, different type of amino acids. As an example, an amino acid, here's amino acid 1, amino acid 2, and what you form the polymer by taking this carboxylic acid group and with this amine group and condensing them to form an amide bond. And so you grow a protein or a peptide by making multiple peptide bonds like that, using different amino acids. So insulin, like lots of other proteins, are essentially made of different amino acids. What makes these different peptides different is the sequence of the amino acids in these peptides of proteins. And what makes each amino acid different is in fact this R group, side group as it is called. Okay? Alright, so this was around if you go back to the previous slide, in the 50s, 55 and so forth. In 1953, there was a seminal paper by Watson and Crick that discovered the structure of DNA, the double helical structure of DNA. And it's an amazing, if you were to trace back the biotechnology revolution in the 20th century, you can use that as your starting point, as it were, 1953. Of course, there were lots of discoveries before that and after that, but this is a very, very good time point to look at what happened after that. So what Francis and Crick discovered was the structure of DNA. And along the way, discovered how does DNA carry the information which allows it to make copies of itself. And then later, scientists discovered how to make different proteins, for example, from the information that is coded in the DNA and so forth. So fascinating discovery in 1953. And in a few years after 53, scientists also discovered the genetic code. And what they discovered was 3 groups of nucleotides. So for example here, A, T, and G, if they're together, it codes for a particular amino acid. So in this particular case, ATG will code for one amino acid, CTA will code for some other amino acid. So it turns out that the sequence essentially is information. A 3 nucleotide sequence together codes for one particular amino acid. So you can imagine if you want to code for insulin, all you had to know, if you knew the insulin amino acids, you can imagine getting the DNA which actually codes for those amino acids, because we know the relationship between the amino acid and the 3 nucleotide groups together, or what's called the codon. So it's an amazing time. So we were able to, we can actually design DNA sequences that correspond to specific protein sequences. And so this is just the genetic code in an easy way to read. As you can see, if it is C, for example, you have UCC is codes for serine, UCA codes for serine, and so forth. So using this table, you can map the 3 codon sequence to the amino acid that actually codes for. And so the other thing that also happened around this time is chemists discovered efficient ways of synthesizing nucleotides chained together. So just because we happen to know the sequence doesn't necessarily mean we've solved the problem. We had to find efficient ways of chemically synthesizing them in the lab so that we can engineer production processes. And so the chemists came out with efficient ways of making nucleotide synthesis. And here is an example. There are two different types of nucleotides. AT is one type, CG is another kind. Purines and pyrimidines, as they're called. And the chemists learned to work with them and make efficient synthetic processes using them. Right? Okay, so the significance of the discovery since 53 was that we knew the relationship with the triplet codon and amino acid, and we figured out a way to make synthetic DNA. By that I mean take individual nucleotides and chain them together to make polymers to make synthetic DNA. That is an amazing and important point in the biotechnology revolution. In addition to that, we also discovered that nature has a way of making this DNA in the cells. So instead of the chemists doing this in the lab, for example, biologists discovered enzymes, proteins, that could do this chemical synthesis very efficiently inside the cell. So we discovered enzymes. And we could purify them and therefore use them in a process outside of the cell. So we could do amazing engineering. Right? We also discovered, biologists, with really primarily work of biologists, discovered how bacteria make proteins, DNA, enzymes, and so forth. That is, we started figuring out how bacteria work. What is the relationship of the DNA to the proteins, and the proteins to the DNA, and so forth. So we know a great deal about that today. So sometime in 1978, Genentech was the first company that actually synthesized a gene or a sequence of nucleotides which codes for the insulin molecule. And then they engineered a bacterium to express that particular protein. Imagine, insulin was discovered in pancreatic cells. Frederick Sanger discovered the sequence. And with the biotechnology revolution, we're now able to synthesize the gene and have a bacterium make the gene that was originally made only in the isolates of the Langerhans and pancreas. So we are now able to do something in a laboratory that used to be made in organ systems. So it's pretty amazing. So they synthesized the gene for the alpha and the beta chain of insulin. They package the gene into plasmids. Plasmids are essentially a collection of genes and some additional material to make those genes functional. Put it into bacteria, and then the bacteria, in this case E. coli, was induced to make the insulin protein. This is the Genentech's work. So it was pretty amazing. But here was an important problem. They could make the bacteria make the insulin protein. Then they had to make large quantities of insulin in large fermenters. So that took some engineering of its own, because you now had to deal with hundreds of thousands of liters of bacteria that had to grow carefully, and from which you had to recover the insulin. And of course, then purify the insulin. That's also going to be extraordinarily important. And then this last slide, like I did in the slide for penicillin, this is an example from a software called SuperPro from a company called Intelligent of an insulin process flow diagram. Again, don't worry about the details, except this is an example of what a process would look like and process actually used to make insulin, containing different operations, different unit operations, including mixing, reaction, filtration, different chromatographic methods for purification, and so on and so forth. So this is an example of the engineering that is required and design in order to make the product, in this particular case, insulin.